Dr. Richard Roberts. And so Dr. Richard Roberts graduated with honors and highest distinction in chemistry from the University of Kansas. He then received his PhD in biophysical chemistry, working in Don Crowther's lab at Yale. There, he was the first to establish the striking stability of RNA-DNA hybrid triplexes, determine the specificity of triplex recognition, and develop a set of thermodynamic and kinetic rules for the formation of nucleic acid triple helices. In 2006, he joined the faculty of USC, where he is an associate professor of chemistry, biology, and chemical engineering. Please welcome Dr. Roberts.
Hi there, everybody. Can everybody hear me okay?
Yes.
Yes.
Yay! All right. I just want to thank the organizers. I thank Katie for getting me involved in this, and I thank all of you. I mean, I guess I would say that I'm extremely excited. I'm extremely excited about the community basically sort of getting out and talking to people and finding out how we can sort of improve the health outcomes of the whole community and improve the lives of people by vaccinating them. I am super excited about that. I was asked to talk a little bit about how the RNA or how the vaccines work. And so I've got like just a few slides in PowerPoint, which is always deadly to sort of go over that. But I also think I guess I'll just say like my own experience in interacting with people on this is and even some USC faculty, just trying to find out what people actually are concerned about and sort of what their worries are and what their fears are about the vaccines. And I think a lot of it seems to be that people don't necessarily really understand how the immune system kind of works. And I mean, that's okay, because the immune system is very complicated. We definitely don't know all of how it works even right now. But there's some really basic things, and I'll try to cover some of those in just a second here. Okay. That work? Yay. All right. All right. So right now we've got 3 approved vaccines, and they all kind of work in the same way. There's the 2 that we have used the most are brand new kinds of vaccines. They're called messenger RNA nanoparticle vaccines. But what they really do, all of them do, is kind of the same. And what they all do is they basically find a way to generate some protein called spike. So this is this image here is a picture. It's an image, a mock-up of what we think the virus looks like. Of course, it's not actually colored, it's just colorless, but it's very pretty and looks good. And these little pink doohickeys here are this protein, which we call spike. And the key, and all of the vaccines to date so far are directed at making antibodies and T cells that target the spike protein. And so why do we care about spike? So one reason we care about it is because without spike, coronavirus, this coronavirus SARS cannot get into cells. So, like everything else, it needs a handle to grab a hold of to actually get inside. And so the spike protein, its main role in the virus is actually to grab a hold of, to physically reach out and grab a hold of proteins that are present in your body that are on the surface of your cells. And it grabs a particular protein called ACE2. And one of the things people have said to me about vaccine hesitancy is they're like, well, how did we develop this stuff so fast? And the answer is new technologies basically allowed us to determine the sequence of the coronavirus in a matter of days from the first time that we noticed it. And we realized immediately that this virus was actually very similar to a virus that we had seen about 17 years earlier that we called the first SARS-CoV, now SARS-CoV-1. And so that's kind of thing one to know. We immediately recognized it as something very similar to a problem we had before. And the second thing is, we've been studying SARS viruses, these coronaviruses, for a long time. And that's how we know that the spike protein is so essential for the virus to get into the cell. Like we know exactly where to target because we've studied this protein, we've studied this virus for actually quite a long time. Now, the novel coronavirus SARS-CoV-2 is a little different. It's a little different, has some new properties, but it's really similar to in the way it behaves with other things that we already know a lot about. And so that's the first thing to know. We really, in January of 2020, we knew already then what we had to go after. And all of the work that took place over the next year, the furious work that took place to get these vaccines made, basically leveraged off of that initial knowledge that we essentially had built from then and from the 17 years prior to that. So I guess the second thing is, how does your immune system work and how do you get your immune system to be ready for an infection? And you can be infected by the virus and that'll generate an immune response. But the way your immune system is fascinating. You have roughly 1 billion different sort of widgets, and they're all kind of different shapes. They're antibodies and T cell receptors. And there's so many different shapes that they can recognize kind of almost anything. And that's how we can recognize new viruses that we've never seen before, new infections, new problems that our immune system has never been exposed to. The problem is that, we have a lot of different things, but we don't have very much of any one of those things. So there's in each of us, there's a few different antibodies that recognize SARS-CoV-2, the spike protein. But there's only a few of the cells that make those. And there's only a few of the T cells that would recognize those peptides on the surface of a cell. And so it actually is going to take some time. Cells take about a day to divide. And so when you get infected, your immune system takes some time to build up resistance, a couple of weeks or 3 weeks to actually get that resistance up. Meanwhile, the virus replicates much more rapidly than that. And so that's why when you first get infected, you get sick, because the virus basically replicates rapidly and you just have a few people defending the fort. So really when you're immunized, or what you're doing with immunization is you're training your immune system. You're basically going from a few things in one area to many, from a 10 to maybe 100 to tens of thousands or more cells that recognize. And so when you're attacked, your immune system is already ready to go. It has B cells to make antibodies and T cells to kill any infected cell. But it takes time for that to happen. And that's why we do a vaccination and then essentially a second vaccination, a boost, and then essentially a follow-up, which is what we're doing now. So how does this actually work in the cell, and how do these vaccines actually work? It's quite cool. We basically take the instructions for making spike. So CoV-2 has 14 proteins. We take the instructions for making spike, and we basically wrap them up in a little fat ball. We call it a lipid nanoparticle, but this little fat ball basically enables that RNA to fuse with cell membranes and go inside the cell. And then this instruction basically tells the cell to make spike protein. It makes some of it and presents some of it on its surface, and that excites T cells and other cells of the immune system. And then they go and run around, they propagate and run around and kill any cell that is expressing this foreign protein. And then little bits of the virus protein leak out of the cell and probably stimulate B cells, and these make antibodies. And the antibodies now are neutralizing. They'll coat all of the spike protein and basically make the virus so that it can't infect any cells. And I guess a couple things here. One is the instructions for this are very readily broken down. So there's no chance of making any changes to your own genetics. It just is a way of telling yourself for a short time to make some of the spike proteins so that you get an immune response. The second is the only protein that we're putting in there is spike. SARS-CoV-2 has about 14 to 16 other proteins that are necessary for making a virus particle. We're only putting in just that one that we need to get an immune response to that will block the infection and allow us to kill cells that are infected. So that's the basis of this. The other vaccine, the J&J vaccine, instead of being a lipid nanoparticle, is just a different type of virus that basically doesn't do any damage and doesn't replicate. But it does kind of the same thing. It delivers a nucleic acid package that instructs the cell to make some protein, and you get a T cell and B cell response.